Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa. 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 176th seminar, and I call it a University College London Day. I am attending the Biomedical Engineering Society meeting for the first time here in San Diego to give a talk and also serve as a panelist. It is so exciting to meet new colleagues in a conference where I am actually a newbie in this conference. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Leo Rios. He is a professor at biochemical engineering in University College London, or in short, UCL. I got to know him at a social media platform in 2021 when I was also a newbie because I just started social media engagement for the first time in 2021. Recently, he kindly hosted my seminar at his university and gave a few hours of walking tour in London, including authentic British breakfast. He is one of the kindest persons I ever know, and he kindly accepted my invitation to give a talk for this SynBYSS audience. Leo, thank you so much for your contribution to the community and also again for your hospitality in London. And the virtual podium is all yours now. And thank you again.
Thank you. Thank you. Thanks a lot. Then Taksun, it's a pleasure exactly to be here. Been a pleasure exactly we met in a few conference Taksun and very nice and to see you now exactly, be involved in this amazing series of seminars that have a huge impact, in our community. And exactly also lovely to now meet you also, Gita, because our research is very similar as we're gonna see. And I prepared just a quick presentation just to show a little bit of what I'm doing in my research, but exactly, I mean, really quick to connect a little bit then with some of the tools that Gita will talk a bit later on. So let me just share it here and share, yeah. So yeah, just to confirm, you can see my screen, isn't it nicely
Can
there?
you flip in that way? We do not see the
Ah,
your yes.
yes, the opposite way. Okay, let me just see. Okay, yeah. Maybe, or actually, one second, I will just share the PowerPoint instead of the screen.
Mm-hmm.
So share PowerPoint, share. Okay, and then that should be
Wonderful.
excellent.
Thank you.
Okay, perfect. So yeah, I'm a Leonardo Rios, as you said, based at associate professor at the UCL and biochemical engineering. Exactly. This is here we're based in London, as the pictures look like a really sunny place. Now it's actually not so bad. And it's an interesting campus, not like a big department, around 25 academics, but all focus on this biochemical engineering, where a lot of the synthetic biology or engineering biology has a big impact when we're more in that application side, like this interface between bioprocess engineering and engineering biology. In our case, our research is heavily linked to industry. Lots of research, I don't know, the majority is with the industry. And so we cover these both aspects, like, yeah, the scaling up, we really try to tackle some of the problems that engineering biology is facing in the scale up. But then also a lot of the scale down, really also taking advantage of those fascinating micro scale tools that have been, used to accelerate engineering biology for not only for DNA assembly, biofoundries, but also, for example, we're trying to push to create what would be more like a bioprocess biofoundry, like connecting that steps. That's something may not talk much in this case, but we're pushing forward, maybe hopefully join the biofoundry islands, but to give, how you call it, alliances, but to complement that, because I think that's alleviating some of the bottlenecks for engineering biology. So this is a little bit then some of the things that my research group does, so it's this metabolic engineering. There is the automation, but as I said, pushing a lot more towards bioprocessing, this biofoundry, and for example, continuous flow things, or how, this bioprocessing AI, for example, it's also having a lot, and how we can integrate. The key thing is integrating this, how to design those microbial cell factories, but taking into account also the next also bioprocessing levels. And okay, one of the, in this case, I decided to talk a lot more where we work a lot more in my group, which was, for example, an example of making some of those molecules, pharmaceutical molecules, anti-cancer drug, Taxol from plants, high value from plants, but it's a little bit still more expensive, not sustainable. So that's where yeast is one of those microorganisms that we believe we have really nice tools, and Gita will talk us more about that, because it's a complex pathway that really then we really need very fine tools to really try to be able to produce it in a heterologous way. How we do that? So then, going quick, we really down. This is when we need a lot of the genome scale models that other groups have developed. But then this is key thing, this whole bioinformatics, really nice tools and the toolkits, lots of promoters, lots of different types of, to engineer, let's say, like targeting different organelles, fancy tools like regulation, and to engineer those cells in a quicker way too. And then also something that we've been pioneering is using some bioreactors, but that they mimic larger scale. Something that we thought is that all of those, people were testing it in plate readers or in shake flasks that never really mimic a larger scale. So no, so been pushing to do micro scale bioreactors and mimic. So that's a little bit what we're being working. I'm switching a little bit. What do we usually get in our type of data is, with the genome scale models, yeah, we can get now, especially because it's quite advanced for Cerevisiae, the organism. So we can, use these informatics tools to, in a rational way, design the metabolism. And then with the microscale tools and with SynBio, we can generate a lot of different mutants that we can start comparing, like with the microscale tools are comparing which one is kind of the best. Uh, but not only comparing, like the production, like in a shake flasks, but also we want to compare how do they start to behave in a bioreactor. So this is microscale bioreactors. We're checking, what's happening with the pH, what's happening with the oxygen, or the effects of those, where we're engineering those cells, and seeing sometimes, that they're all tested, in a shake flask, but like you're missing that the oxygen is totally different, the pH is not controlled. So we're really combining that. And at the right, we have what we call metabolomics, trying to measure all the metabolites. So again, I'm going quick, but we're really trying to incorporate all that. And I think when engineering then say, microbial cell factories, taking all that in consideration is important. And exactly, those tools that Cerevisiae has been fantastic for do that. Now, something that many times that we have also noticed that the purification sometimes is a bottleneck that we have totally downstream processing overlook in metabolic engineering. So we've been working in, for example, those Taxol components, they need to be very pure. And so we've been really working, for example, here we're using very specifically designed beads, materials that can absorb all those different type of metabolites, and then not only can help us to purify better, but sometimes, uh, for example, I'm going to go to the next slide, but sometimes we can even detect compounds in very low concentration that wouldn't even be able to sometimes detect with other compounds. So this is quite important, especially if it's, for pharmaceutical products, purity is quite important. And but that's actually going beyond, it can really help sometimes to make bulk chemicals, maybe, like a lot cheaper, the purification, you don't need to break the cells many times. Or so we're also been pioneering in some of those systems, and then you can do it in a continuous flow system to do it like that. This is more, with those systems, we can do continuous fermentations. So you can keep running a fermentation, continuously removing the products with those beads. So it enables, instead of a batch fermentation of 2 days, you can keep running it, for 10, 15 days. So then and we're showing, like then it's a lot better, the semi-continuous or continuous compared to batch. We can purify, all those different components. We can purify in different beads. And then, we can start purifying, like different components and then making it a lot cheaper, or further analysis, or testing all those components with these different types of materials that we're using in this case. Now, those tools, interesting, we developed for yeast. You wanted to touch us for some of that, with collaborators. They have approach that we can use the same for plant cells. Plant cells that make taxol. We could also engineer, and that's a cellular, plant cell culture, we can engineer them, trying to make different taxols and using, for example, those bits too. It's also working very nice. That's another collaboration. And that's interesting, some of those interfaces, that has been quite nice. Here I'm just showing when we're working with the plant cells. This is the control, let's say in the bottom left, like how much taxol is being produced. With those bits, you can remove the taxol. Sometimes the product can be toxic. Sometimes you're pushing the cells to make more than that one. Those are some of the cells can be engineered to produce more, and it has released the product, so that was also a very successful approach. And just to finalize, also maybe something that we probably you've seen also, Gita, when we're engineering the cells, many times that's what I'm pushing to also do the early scale up. The cells get stressed easily when you're scaling up, and they behave totally different when you have all those different when you're mixing a larger scales, there is a little bit of fluctuations in pH, oxygen, temperature. That makes them behave totally different. In this case, they start to become pseudo-hyphae formation. Big problems. So how to detect that? Now we can engineer those cells nowadays. Before, we had to find the best conditions to avoid that in the bioreactor. Nowadays, we can actually genetically engineer those cells, Yarowiya, political, so you said, to avoid those scale-up issues. So it's been fantastic how the engineering biologists were even managing to make more robust cells uh, for industrial scale conditions. So this has been in the last few years, that's been amazing. And now, we can engineer cells exactly to, as I said, avoid that. Another approach that we have tried in my group was the how you call, like consortia, dividing the pathway, Escherichia coli. Yes, that could be another one. We've done a little bit of work into that one. It's also something that we believe could be a solution, for especially doing more complex products. And in our case, it was fantastic. For example, we divided this taxol pathway. In this case, it was actually only four different yeast cells. In this case, it could also be yeast E. coli. And that was a really good way to produce different molecules and do this labor, or the division of labor in a successful way, and which is one of the approaches, that we had our recent publication and doing that one, and I'm just saying a little bit, okay, a little bit of the discovery of the enzymes, that we use in this pathway. We'll skip a little bit that one, but then exactly, not just saying this one was a very successful also with the microbial consortia. Now, something to finalize is now with AI is really changing the way we're doing things, So, for example, we are one of the first that our system, our bioreactors, we're making all the data in the pilot plant goes to the cloud. So we, instead of just doing some quick optimization, we're pushing to do really quickly the digital twins. All the data stored there could be shared. And with artificial intelligence, for different projects, a lot quicker to have those digital twins exactly made it. So the next optimization you do, you're not starting from scratch. You can do it a lot quicker. Nowadays, you only have that for the bioreactor optimization, but we're really trying to push also to include also more engineering biology, like data from engineering biology. So that would be what we're pushing, which we believe will be the future also, And so this is some of, how it looks like connecting into the cloud. We're working with NVIDIA, to help us to do that. So it's been a really nice collaboration with them. And the idea is exactly, to make it really quickly, those digital twins. We can then the AI will optimize the bioreactors. Everything I says in the cloud to, for people to share working with that data, and AI also to design and optimize the next process. So that's something we're pushing a little bit. So ideally, we just don't want now to get, like we want to give quickly, a virtual model of the whole process. And not only for, as I said, in this case, it's only bioreactor, but then in the future, we think also hopefully to engineer the cells, So then that's what we're pushing for. So just finalizing, just saying that, and well, maybe I didn't mention a little bit about that, we did a lot of some automation to get all of this data, let's say, to engineer. And that fits really beautiful with what I talk about AI, because then, if you can gather all that information, and it's totally multidisciplinary. And then the way we're doing that, and I think this has been key, these collaborations to do that. And now I talk only in a pharmaceutical taxol, but this is having a huge impact in totally new fields like the food, energy, and other areas. So I'm really excited to see how this is totally changing the world. And yep, happy to end in that. Thank you very much.
Thank you so much. This is excellent. As you know, I mean, we have almost 35 years of metabolic engineering history, and I believe this is now time to deliver real product to the market. And then what you are doing is very important because even if we can produce a lot, purification, separation is another important thing. And then now you're combining AI with the metabolic engineering to really make the huge impact in the world. So I really, impressed by your work for many years, but now also you kind of summarize nicely today about your vision and then what you are doing. So this is an excellent presentation, and thank you so much again.
Thanks a lot, Asmund. Yeah, thanks a lot. And I totally agree with you. Yes.
Sure. Okay. Now the main speaker of today with our longer introduction. Professor Gita Naseri is a synthetic biologist, currently leading her own independent research group at Humboldt University of Berlin. Her academic journey began in Germany, where she earned her PhD from the University of Potsdam, followed by postdoctoral research at Humboldt University and of course, in Emmanuel Charpentier's lab at the Max Planck unit for the science of pathogens. This week is actually Nobel Prize week, so this is kind of right moment to have her in this seminar series. Gita's research explores the transformative potential of synthetic biology to deepen our understanding of microbial cell function and reprogram biological systems for targeted applications. Her work aims to unlock new possibilities in healthcare and environmental sustainability, recognizing that the design, build, test, run cycle is often a slow process, even though we use, automation. She has developed cutting-edge synthetic biology tools such as programmable synthetic transcription factors and also combinatorial optimization method to speed up this cycle and modernize bioengineering projects. She is also dedicated to fostering interdisciplinary collaboration and pushing the boundaries of what is possible in synthetic biology. One personal note, I just today got to know she is now a mom of the two twin baby, now 4 months old. And I always wanted to say, mom is Wonder Woman, and working mom is super Wonder Woman, and she kind of working hard at the same time raising the most important human, uh, two babies, and that is amazing thing to me always. So Gita, thanks so much for your time today. And please take it away. That's all yours now. Thank you.
Thank you for your kind introduction. And also thank you for having me here today. Can you see my screen?
This is coming up. Yes, wonderful. Thank you.
Yeah. Well, we are junior microbial synthetic biology group based in heart of Berlin, Mitte, and my labs are located in Humboldt University of Berlin and also Max Planck Unit for the Science of Pathogen in Berlin. Well, we are interested in application of synthetic biology to understand first cell function and then to reprogram it for desired output. For example, production of a high-value natural product in heterologous house microbial cell factory that they cannot produce it naturally. And like all engineering project fields, we also need some tools for our bioengineering projects. And we focus on tools that enables, like inducible programmable tools that enables us to separate biomass accumulation from production phases. And also genome editing to add a massive genetic modification into the genome of our target organism. And recently started to establish artificial microbial community to divide the complex task between the cells. And then, for example, if you want to build a car, once you have the pieces, then they need to be assembled together to build the car. And in bioengineering project, we also developed some assembly methods that allows us to rapidly make complex genetic circuits. And then for sure, the car needs to be tested for its function. And in this context, we also developed biosensor that enables to link productivity of the system with easily detectable output like fluorescent signal. And also we integrate rapid sequencing technology to our platforms that enable us to speed up data-driven design of new bioengineering systems, for example, car with improved function. And then this toolbox that we developed, we implement for two different applications. First, optimize synthesis of like rare natural products or biomaterial like leather alternatives. And also we use them to understand complex gene regulatory networks. But today I will mainly focus on first application and natural products. Well, there is growing interest in using natural products for various applications, like to use them as a biopesticide or for pharmaceutical drugs. Natural products represent over 50% of drug in clinical use, and indole alkaloids are one of the largest family of natural products produced by plants. For example, and they are used for various type of pharmaceutical applications. For example, vinblastine can be used as an anti-cancer drug to treat different type of cancers. However, the extraction from nature is not efficient. For example, to have 1 gram vinblastine, 530 kilogram of dried leaves of Catharanthus roses, a little flowering plant is needed. And also the chemical synthesis is challenging, mainly due to the very complicated structures. So we need solution for sustainable synthesis of natural products. And here you can see the long biosynthetic pathways that are usually involved to make such a complex natural products in plants. And yeast, in principle, can be used to produce this type of natural product, mainly indole alkaloids, because produce the required precursors. And in 22, there was a group who could successfully express 36 enzymes for production of vinblastine in yeast, and they could produce two required precursors, but in very low level. And this is mainly because this type of natural product can be toxic for microbial cell factory. And also, they could not have active form of the last enzyme P450 that is needed to convert these two precursors to vinblastines. Indeed, P450 enzymes are key player for many enzymatic steps of alkaloid biosynthetic pathways. And yeast Pichia the non-conventional yeast, seems to have a higher conversion rate for this enzyme, 70% higher than Saccharomyces, and it has high expression capacity for non-native proteins. However, there are limited engineering tools for this one compared to Saccharomyces. And what else makes yeasts in general a suitable host for expression of this type of enzymes is like the compartmentalization that has like exactly how plant have it. For example, it has peroxisome that can be used to target acetyl coenzyme I conversion to GPP required precursor for alkaloid production. Also, it was shown that several enzymes for vinblastine production can function efficiently within the peroxisome. And also, it can be targeted to express cytochrome P450 enzyme that needs its a membrane-bound redox partner that can be somehow anchored in endoplasmic reticulum, nearby organelle. And also the acidic pH that vacuole has is a problem, make a suitable environment to produce one intermediate of such an alkaloid metabolite. However, the design, build, test, learn cycle of such a complex bioengineering project usually is very slow. And our goal is to shorten this cycle by establishing a comprehensive toolbox for rapid engineering of long biosynthetic pathways in yeast. Indeed, we try to develop inducible system that enables enzyme expression after growth phase of the cells. We also by using a high throughput assembly method, we try to generate library of a strain like million within a month. And why is this important? Because when we talk about natural product like alkaloid, they can be toxic for chemical cells. And usually making one strain, we need to make many strains until having one that can have enough output for our target metabolites. And also, by using biosensor, we can rapidly quantify the productivity of each member of the library to an easily detectable output signal like fluorescent. And by real-time bioprocess optimization, we can also address the poor reproducibility of bioengineered system. But what I will focus today is mainly on the first two tools. And yeah, the first one, artificial transcription factors. The common practice in metabolic engineering project is like to use host native promoters to express pathway enzyme. However, they are not many, they are most of the time constitutive, and they are not orthogonal. So in the past, we developed a collection of chemical inducible artificial transcription factors for to enable gene expression in various organisms from Saccharomyces to Pichia pastoris and also bacterial system like E. coli and Salmonella. And in our collection, for example, 30% of this ATF seems to be, they are up to tenfold stronger than, for example, yeast constitutive TDH3 promoters. And just a little more about this, the structure of these chemical inducible artificial transcription factors, mainly plant-derived. Actually, this artificial transcription factor have combination of various DNA binding domain that the majority of our members of our collection have plant-specific transcription factor DNA binding domain, or they can also be from, for example, DCAS9 or tail. And then they have host compatible activation domain that can be yeast, Gulf4 activation domain, can be bacterial activation domain like SOX-S, or can be VP64, the common one. And then these ATF are located downstream of a promoter that can be targeted by an inhibitor, for example, LAC-I. And when we have chemical inducer like IPTG in case of LAC-I, then this ATF can be expressed, and then it can target a binding site of ATF that is located in a synthetic promoter upstream of the gene of interest, here fluorescent protein. And then we can have expression of the target protein. What else I would like to mention, usually when we design such an ATF for bacterial system, it's very important to mention that it's highly sensitive to the position that ATF binding site is located. As you can see, Dang and co-workers could show that there is a narrow window in a strand and in template and non-template strand of promoter that can be used to target the ATFs. and otherwise we lose their activity a lot. That is not the case that is less sensitive in case of, like, for example, yeast. And we also implemented this principle when we were designing our ATF for bacteria. In the case of ATF for bacteria, we, for example, made them arabinose inducible. And yeah, here I show you one application for this ATF, for example, production of beta-carotene in yeasts like Saccharomyces and Pichia. And to produce beta-carotene using our ATF, we need to express 3 genes. And also we used our ATF to redirect yeast endogenous pathway to enhance production of FPP, the precursor for beta-carotene synthesis. And we could show that that this strategy could enhance production of beta-carotene in both Pichia pastoris and Saccharomyces. The good point for these ATFs is that, like, they are very versatile. You can use them in bacterial system, you can use them in yeast system, you just need to adopt the activation domain. And what we are currently doing is to improve the gene expression dynamic. For example, we try to make them responsive to temperature and also making light responsive ATF. And yeah, when we go to a strain construction, for example, using this ATF to express pathway enzyme, usually it's very time-consuming procedure. And if you generate a strain that cannot produce your target metabolite in sufficient level, usually you have to be back to your strain to change the promoter of the genes, sometimes to use a stronger promoter, sometimes weaker promoter. That can be very time-consuming using classical methods. And to address this, we developed a method that we called it Compass Combinatorial Optimization that allows us to generate a library of yeast strains between million members in just a month. And just briefly to tell you how does Compass Combinatorial Optimization cloning works. For example, you can have a collection of ATF as shown here, like 9 ATFs, and that you can add them to your cloning reaction that together with your gene of interest and the backbone plasmids. And as all ATFs have same overlap region, so you should be able to get all of them assembled upstream of your gene of interest with the same equal chance. And to make this method independent of sequencing, if you add the promoter of an antibiotic selection marker, for example, to your inserts, and if you have the coding sequence of this antibiotic in your backbone plasmid, then you should be able, by selection on selective media, here like ampicillin, to just get those constructs that have been correctly assembled. This helps you to avoid like sequencing. And you can even further extend this concept by, for example, having more genes and more backbone plasmid in your cloning cocktail. And then following E. coli transformation, you can split the transformed cell, for example, in this case, 2-3, and then plate them on different E. coli plate, LB plate with different selection marker. And so then this will allows you, if you have 9 ATF, to be able to get 27 different plasmid just using one cloning reaction. And when you build such a transcriptional unit, actually you are in level 1, and then this transcriptional unit can be directly integrated into the genomic site of yeast, multiple sites, or you can assemble them in larger plasmid and then integrate them in yeast, in like sites, 2 different yeast sites, like that also somehow give you positive selection possibility, like ADE2 and LIP1. Using this method, we could make Narion library that are co-producer for naringenin and beta-yonon. And actually, we have the biosensor that can detect naringenin, and also beta-yonon is colorful product, downstream product of beta-carotene. And then we could show that only by checking 10 to the power of -6 of theoretical complexity of our library, we were able to have a strain that could produce 4.2-fold beta-yonon higher than what was reported as a high producer at that time. And what I like to mention here when we talk about combinatorial optimization, always the tricky part is like you need to have a good screening method to be able to quickly screen the producer within the library. And when we talk about alkaloid, fortunately, there are some biosensors for this. For example, a Linton lab developed a collection of repressor mutants from bacterial system that can sense alkaloids. And like indole, they did it for different type of alkaloid that highly possible to also found one from this collection that can also sense our indole alkaloids. And if we could find a mutant that can sense our alkaloid, then you will be able to get like fluorescence signal. And then if you engineer your library with this biosensor that we are currently working on it, then you should be able to rapidly can detect the strain that can produce like high amount of your chemical, because then you can sort the cell, for example, flow cytometry. And yeah, what I would like also to mention is like our goal, as I told, is to develop comprehensive toolbox that we want to use it for long pathway, engineering life pathway, not only the natural product that have pharmaceutical property, for example, those that can be used as a biopesticides. And then this toolbox can also be used, for example, for other purposes, like in basic science, to study complex gene regulatory networks. And this already brings me to the last of my presentation, that I would like to thank to my team and also the funding, and also for you for your attention.
Fantastic. Thank you so much. I want to ask whether Leo has any question or comment.
Yeah, no, that was a fantastic talk, Gita. We should actually discuss later on. I was quite interested with especially those exactly those transcription factors, that you have developed. Are those mainly is it Cerevisiae or is it for Pichia that you have them, or is it or E. coli? Or yeah, that's also, I was not totally sure. I mean, is it
Yeah.
both Cerevisiae and Pichia? Isn't it? Okay, yeah.
Yeah, for all of them. The collection is bigger for sure. For Saccharomyces, we have 106 transcription factor, but we also tested some of them in others, like 20 of them in Pichia, and some in E. coli, in Salmonella, and other gram-negative bacteria. And by changing the activation domain, usually we get good expression range for them. Yeah, in principle you can. And right now we also try to bring them to filamentous fungi. To be honest, they seems to be compatible, because the only thing is the DNA binding domain need to be orthogonal. That is from mainly plant-specific transcription factors, and you just then need to adopt the activation domain. And yeah, so far we are happy with them. And the good point is like they are not like CRISPR-based ATF. They are not big. No, they're ATF. These are smaller.
And then, for example, but then, okay, so those are for specific chemicals, from probably plants, isn't it, that have, um, how would you approach it? Let's say we have, imagine, okay, I have a molecule, could be also from plants, that I would love to have this transcription factor. Or would people have tried to use, your library to maybe find the closest one that would work for, like a molecule that we may not have a transcription or factor? And could we use this to try to, maybe discover or find like a transcription factor that, like could help us, to, like a different molecule than the one you have there, or or how would you approach that?
I'm not sure if I got the question. You want
Because
to
for
use
example, this collection is each transcription factor will be responding to a specific chemical molecule, isn't it? That
this transcription factor, for example, here you see, yeah, it depends which type of responsive chemical inducer you want to use. If you put, for example, lacoside in the promoter that encodes this transcription factor, then you have like IPTG responsive transcription factor, right? If you use like, or you can also, we also did like with arabinose, and now we are doing with light.
but could I use that to exactly to okay, but not with other type of molecules yet, isn't it? Okay, like if we wanted to respond to what,
Uh-huh.
not yet, isn't it? So this
Not yet.
so this is for a specific molecules, but then you can put it exactly in your promoters and you can control and then when you said also that like where you're gonna put in, upstream in the sequence was really important. Is that species dependent, or it depends definitely of the transcription factor you're using and the promoter you're using, or is it something a little bit more general, that depending of this, or how general is that, or is very specific for each combination?
To be honest, you mean you are talking about the strength of the ATFs, like how much strong they can be and can be predict.
And
To
when
be
you said that
honest,
it's sweet spot, yeah, when you're putting it, yeah.
yeah, when we build, we usually use different DNA binding for domain from plant here. And it's not possible to predict which plant TF is better. Some, for example, when we make collection in bacterial system or in yeast system, always the plant transcription that factor that they call it JUP1 always work perfectly. I cannot explain why. Or there is another one like ATAF1 that usually work in different hosts. And what I can say you, we can always, of course, play with the binding site copy number of the transcription factor that we put upstream of our gene of interest, here fluorescent protein. Not necessarily increasing the copy number of binding site give us higher output for fluorescent protein or other genes of interest. Most of the times I myself, because I did this in the past, and usually with two copy of the binding site, I was getting high expression rate. But sometimes for some ATFs, we have more copy binding site number. In yeast system, we have everything is easier. But when we come to bacterial system, always we have more limitation because, for example, as I told you, this binding site copy number that we play with them, sometimes we make them even 8 copies. In bacterial system, we cannot make it 8 copy because then you will exceed this narrow window that you have for optimal
Mhm.
targeting. Like this is like the work that was
Exactly, yeah,
done.
that one.
Yeah.
Mhm.
Yeah, and we also had the same issue. So most of the time, with 2 copy number of the binding site for our ATF, we could get the best output. But in this system, usually is more flexible. We have even 8 copy, and still we could get increased output. And sometimes orientation of the transcription factor and activation domain change the output of the system, C terminal or N terminal. But I never went to like bioinformatics, like computational analysis to find out how this can, if we can, I can find any correlation. When we find a strong one, we are very happy. But
Yeah.
yeah,
Of course.
yeah,
Okay. No, perfect. Fascinating. But exactly. Then not so easy because you need to do the testing, and that's why your platform that you have developed is very useful, for that.
yeah. What I can say you, for example, now we are using these ATFs to express some genes, and we try to compare in yeast, and we try to Saccharomyces, and we try to compare them with, for example, a strong promoter of yeast, and we get much more fold induction with for transcription with our ATFs. So and it seems they are working quite like nicely. And of course, the integration site is also important, right?
Yeah, totally,
And
totally.
yeah, yeah.
And maybe we have a bit of time. Final question. I was just a bit curious because I was a bit surprised with that first slide. That was a bit far when you said that in Pichia pastoris, the P450s have 70% more activity than Cerevisiae. Is that
Oh,
I mean, maybe, you didn't mention that. I was just, is that the native P450s, or you're mentioning, heterologous P450s that you would put in Pichia that they would also work a lot better, than in Cerevisiae?
yeah, no, I think this the group they discussed about heterologous P450 enzymes expression in PTR.
And that they work a lot, interestingly, a lot more active. Well, that's interesting for
Mm.
what is interesting, okay, such as is. Which one you prefer now, working with the Pichia or with Cerevisiae,
And to
or
be
either?
honest, I know that Pichia has like disadvantages, but still I prefer to work with Saccharomyces because sometimes we found genome editing in Pichia a bit complicated. It doesn't work as we expect. And so, yeah, I would say still I prefer Saccharomyces, although I know that for this natural product, Pichia postoris should be better one. Yeah, and yeah, sometimes some strain they are growing so fast, but then some other strain they are growing so slow. And yeah, yeah, I do not know. Have you
Yeah.
ever worked with PQR?
No, we were starting to work with Pichia, but I agree with the same thing. We are used to Cerevisiae a lot more, and I can understand all it's been a little bit more painful, exactly, than one we expected it, which I wanted to see your opinion, but exactly, we're in the same, exactly.
And yeah, when we have complex natural product cascade many genes, so we prefer to work with organisms that are a bit like easier. But
I agree.
yeah, yeah.
Perfect. No, I agree. Yeah, so no, fantastic. Thanks a lot for the seminar. I'll send you actually a message later on, Gita, we should discuss a little bit more. I have some really good ideas for collaboration we can maybe discuss
Hmm.
later on.
Yeah, yeah. No, yeah, good, very good. Yeah.
Fantastic. I'm more likely bacteria person.
Mm.
But I recently, enter into eukaryotic system, but still to me, Saccharomyces is still difficult for us. So actually we published one paper to develop a sensor in that platform, but we are not thinking of Pichia. Pichia might be much harder for us, I guess. Uh, my question is, the wonderful talk. Also, I noticed you also collaborate with Matteo Kofas and RPI. I believe that's the review paper, if I remember correctly. Uh, but you kind of lay out some of your vision during the talk, also in that paper, I believe. I just wanted to know, I mean, you kind of nicely presented your platform about sensor and combinatorial, approach, etc. I just wanted to see, I mean, in 10 years, I mean, let's say, 2035,
Mm.
what you wanted
Mm.
to see, not only your research, but in the field in general, regarding the sensor and also combinatorial approach. What do you want to see? What is your vision 10 years and even 20 years from now?
Yeah, 20 years. Okay. What at the moment, what I really feel that is missing is like there are many synthetic biology lab, but usually we don't have a standard protocol or tools that we can share together. And actually, what I'm trying to do is like, I like us to see to be there is like to have somehow, I do not know, a platform, a cloud that we can share our standard tools and somehow make them all a standard. For example, this ATF that I'm developing now, our whole goal is like to make them in the way that can be used for different organisms. So then this helps the community a lot to be able, to speed up their work. And if we could do something like this, then something that I think it's missing, like having like database that we can share, like data that we obtain to support AI to
Mhm.
somehow speed up designing engineered system. Because right now I have impression that different labs, they are doing with their collaborators their own project, but somehow it's not like there is not really a platform that we can share our like tools there and then somehow using each other tools more. I mean, we are
Mhm.
doing, but it's not like there are many parallel work going on. And
I see.
yeah, what I think at least I want to try is like to bring somehow the community to make it easier to use tools of other labs.
Mhm. I see. So
Yeah.
that's a very interesting point. This is also kind of question to both of you. I attended a conference in Korea with many big names, including Jay Kiesling, Jim Collins. Also, I'm currently attending a conference in San Diego, and then many of the talk topic was about AI and automation. And I met a lot of people, including Nathan Hilson. He's also in running automation lab or the biofoundry. So the question to you, both of you, Leo nicely, presented at the end of, presentation, big vision about automation and the use of the AI. And then I believe in the future, even 10 years later, we may have fully autonomous, uh,
Mhm.
kind of pipeline, I guess. I just wanted to hear your very short, vision regarding that autonomous lab or automation AI slash kind of the future in biotechnology.
Yeah, like I can go a little bit in that, I mean, uh, I think exactly. I mean, the huge impact of that will be, especially in industry, adopting, many of those techniques. There is a lot more also routine to sometimes to do automation, because sometimes in academia we're always trying to do new things, new way to do things, but the impact that is gonna be having in industry, let's say, for how they are, like also for example, finding, screening new medicines, new products. This is gonna be massive, because those are the ones who are really taking a lot more advantage of those. So the impact, for the speed that we will get new products, but hopefully also new solutions for is gonna be massive. And also academia, I'm really excited because exactly, I think it's really pushing us to. I mean, I mean, before, for example, it would take us a lot more time to do, things, or when suddenly, we got a tool like a calculator, we didn't have to spend much time to. So so it's really pushing the boundary. I don't think it will be like replacing, maybe people get scared, gonna replace a good job. It's gonna not really push a lot more into thinking big. Undergraduates are gonna be able to, like in their classroom to do things that before it would take us, whole load of PhD time to do, So I do see coming that way. Now, it's exciting. It's a little bit also crazy how things are moving faster and faster nowadays thanks to all this. So I'm not sure, we will be able to adapt, let's say, to that, like things are moving. But I do see more the positive, and I think there's some big challenge that, for example, in my case, we're moving like hopefully curing cancer, like complicated that I think thanks to those tools, I hope we will be able to tackle really those complex things with the use of engineering biology, for example.
Absolutely. So interestingly, we got the kit handler with also 18 plate, culture, capability, also all the handling, robot, capability. We got that machine before pandemic. But at the time, my student didn't use that one much. But nowadays, because of all the AI kind of hot topic and the automation hot topic, some of my students start to use more. So now I'm seeing around 20 to 30% of usage of that machine. So I'm really glad. But by the way, I mean, do you have any kind of, comment, Gita, regarding the question I just asked?
I think we will have one day soon that the lab will be empty and researcher will mainly sit in the office and everything will be automated. And so, yeah, we will. And I also don't think that there is something that should have concern, because then we can focus again with support of AI to design better the systems. To be honest, yeah, I'm very looking forward for this direction. That I don't think I think it will happen soon. That
Yeah,
very soon, yeah, we will not be in the lab any longer.
yeah, but that is also important concern because a lot of young people start to worry about whether we're gonna get any job in the future, AI, replace their job. I mean, but I talked to yesterday our plenary speaker, and then my question is, he's more like a doctor is, perspective, how gonna AI kind of change the medical world. And then he also mentioned, you know, now robot and AI gonna do all the diagnose, also even some surgery, potentially, but most of the diagnosis part. But he said, in near future, still doctor will be there, using AI as a tool to help the diagnosis, but still do surgery, etc. So we're gonna have the, sort of kind of intermediate kind of stage, kind of still doctor is available, doctor's job is still there. I guess I'm the same thing for biotechnology. That's my guess. So we're gonna not losing the job for the human, but still kind of utilizing to speed up. That's kind of my hope.
I agree too, and to solve a lot more challenging problems, that before it would be totally impossible for maybe a single person to try to address.
Hmm. Yeah.
I think we will have somehow we have to accept that to work with AI together, like collaborator.
Yeah.
Yeah,
Yeah.
totally true, 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 true,
Yeah.
and to have this massive impact. Yeah.
But I guess this is time to think about the potential benefit and also potential risk of the AI to incorporate into our, field. Otherwise, there will be some, the side effect that kind of more negative than positive. So I guess, that's my comment.
But can we stop this? I mean, if we feel that there are many negative aspects, can we stop this like how it's going now? I
I,
think
I,
now.
I, I think we can because every new technology has been like that, has
Mhm.
positives and negatives. And it really now exactly depends on the job that we can really exactly get together to regulate the better, and maybe agree between the world, I mean, it's not easy. But I think as Taksun said, this is a really time. And especially that's something academics, we can try to at least advocate a lot more, so maybe later on, people, as I said, will be able to start doing on time, and that's kind
Sure.
of our role, but
Yep.
I think it's as any new technology, it has always happened, those the need to exactly put some security, some regulations, yes.
Right. My point is we need to start to discuss more seriously about all the benefit and the pros and cons right now before it's too late. So that's my point.
What do you think, about that? You think you are not very happy with what is going on now?
No, no, no. I'm very excited. I'm very excited with the AI thing. Uh, we should use AI. This is inevitable. So the future will be, basically, we're gonna use the AI a lot. As you can imagine, a lot of people use ChatGPT. So that's the future. But future should be brighter with caution. That's what I want to say because
Hmm.
I'm also kind of editor of 10 journals. The quality of paper improved before and after ChatGPT. That means a lot of people using ChatGPT for the even manuscript.
Yeah,
So
yeah.
it's a kind of improvement, but at the same time, we also think about uh, all the people, including me, non-native speaker, how to express our idea. If we just keep relying on the ChatGPT, that's not gonna good because our student and postdoc should have ability to express in English, their idea clearly rather than always kind of depending on the ChatGPT or other, the platform. So that's kind of what I want to see. So I kind of a little bit discourage my student to use the ChatGPT, but I believe they're still using it.
Yeah,
So
that's
this is something, I was thinking about for many years. And personally, I never use ChatGPT because I worry about me become addicted to the ChatGPT. So I never use it, even though I sign up for the ChatGPT much earlier than other people. So that is something I was thinking about. Well, I'm excited. The future is much brighter, and then we're gonna solve a lot of global problem, including medicine, biotechnology, climate, etc., with help of the AI. And I also use a lot of AI thing, including Rosetta and also AlphaFold in my lab. Before I didn't think about the protein engineering because protein engineering takes a lot of time, and I actually become a robot when I was graduate student picking 20,000 colony myself and pipetting myself. I was robot, but because of that, I didn't want to do protein engineering. But now, my student has AI tool, including ChatGPT, the
a whole.
AlphaFold and Rosetta, and also the liquid handler. So now I'm doing the more protein engineering than before. So I'm very excited regarding that one. But cautiously, we need to go.
I think what also one more point thing is like AI also try to support us to develop more interdisciplinary fields.
Right.
like what you say now, bringing protein engineering to your
Absolutely.
work. Yeah, I think, yeah. And in the end, always we ask this AI to do something for us. Yeah,
Right.
I mean,
Yeah, absolutely. I know yesterday the keynote talk was so amazing because now we are, diagnose something we never find before, even just using the image and so etc. So that's a very, promising, and I was very excited. So that's why I'm asking that question to both of you.
Mm.
Now I see the 4 minutes after 11 Central Time, and let's close, and then we can chat a little bit more if you want. Okay. Thank you all for joining and staying today. We'll meet again on October 14. Please note this is the Tuesday, not the Thursday. The same time and the same Zoom link. Uh, Actually, that is a schedule originally on December 4 because the speaker cannot make it that December time. We'll have Dr. Pablo Rabinovich at DOE, Department of Energy, Biological and Environmental Research, BER, and also Professor George Liu at Rice University. As usual, the follow-up informal chat will occur without recording. Please stay here. If you are interested in chatting with us, I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thank you. And I stop recording.